Hello everyone, welcome to today's lesson with Nursing Insight with Caleb Mlonga. So today we are going to look at a topic entitled HIV Life Cycle, which can also be called replication process of HIV or the pathogenesis of HIV. So before we go in details about the life cycle of HIV, let's just have a brief background of what HIV is so that we can be able to understand what we are talking about. So what is HIV? That will be the first thing that we look at today. What is HIV? It stands for Human Immune Deficiency Virus. So this virus belongs to a group or a family of viruses called retroviruses that cause HIV, that cause AIDS. So these viruses cause AIDS. So now what is AIDS? AIDS stands for Acquired Immune Deficiency Syndrome. Acquired Immune Deficiency Syndrome. And AIDS is a symptomatic stage of HIV infection which progressively impairs the body's cells mediated immunity to infection. So this simply means that AIDS is a symptomatic stage in HIV or of HIV stage in which it alters the immunity of the person, causing immunocompromise to the infection. The immune system will be compromised. So having defined what HIV and AIDS is, now we can look at how it can be transmitted. So HIV can be transmitted using three methods. So HIV can be transmitted using three methods. The first one is sexually. So under sexually, we're saying this is where the virus is transmitted from one person to another through sex. So this mode of transmission can be through oral sex, anal sex, or vaginal sex, which is penetrative. And then not only any type of sex, it is unprotected sex. And then we move to the next mode of transmission, which is parenterally. So when we say parenterally, this is when the blood of an infected person mixes with the blood of another uninfected person, causing the transmission of HIV. So here we are saying this can occur through transmission of blood to an infected person to a person who's not infected. It can occur due to transmission of blood products such as plasma to the patient, donation of organs, and exposure to infected blood to needle pricks or injections. So those are some of the things under parenteral or parenterally method. Then the last one that we have is the vertically mode of transmission. This is a method of transmission which occurs in mother to child transmission, and it can occur during during pregnancy or during childbirth or through breastfeeding. So this method occurs in mother to child and can occur either during pregnancy, during childbirth, or during breastfeeding. So now that we have a background of what HIV is and how it is transmitted, now we can look at the HIV life cycle. So we can start now. So the first stage is the entry stage. So when the HIV enters the body, it targets the CD4 helper cells, a type of T cells that is found in the body. So these T cells are very important in the body because they facilitate immune response to infection. They help the immunity of the body. So once these cells, these HIV viruses go into the body, they target these CD4 helper cells. So once they enter, the first stage I said is the entry stage. So in the entry stage, once the virus enters into the cells, it requires the presence of certain receptors on the cell surface, such as the CD4 receptors and the core receptors such as CCR5 and CXCR4. So here we're saying once the virus enters the body and goes into the bloodstream, for it to enter into the cell of this body, or the cells that are in the body, it will require receptors that should be on the surface of the cell, and these receptors are the CD4 receptors that should be found on top of the surface, on top or the surface of the cell, where there will be attachment of the virus and the cell. So these receptors on the surface of the cell will cause or will facilitate attachment. So this interacts with the protein complexes embedded in the viral envelope. So these receptors on top of the cells will now interact with the proteins that are found in the cells, in the viruses, and these proteins are the ones that we are calling the glycoproteins. So we have in the virus there is GP120 and there is GP41. These are the two glycoproteins that will interact with the cell receptors, which are the CD4 receptors, and they will interact, causing attachment. So when the receptors from HIV, the GP120 bind, they should bind or hold together to the CD4 receptors, which is CCR5 and CD4. That will mean they will bind and fuse together, making one more like attached. These two cells will be now attached. So after binding, fusion, which is a process or result of joining two or more things together of the HIVs and CD4 takes place. So after these cells have binded together, they've come together and fastened together, holding together like that, there will be fusion that will happen of the HIV cell, of the HIV virus and the CD4 cells. So when these two bind, they will it will result into fusion of which we are saying it is just the joining of two or more things together. And then after they fuse, that will now cause the NRA of the HIV virus to now be released in the cytoplasm of the cell. So the first stage is what we're calling the entry, binding, and fusion stage, because the first thing is when the virus enters the body, it attaches, it goes in and finds the receptors in these cells and they bind. The glycoproteins, the GP120 and the CD4 receptors bind, causing binding, and after they fuse, they will release the cytoplasm into the cell. They will release the virus, the viral genome, which is the HIV RNA, into the cytoplasm of the cell. So that's the first stage. So the HIV RNA, which is a single-stranded RNA, is converted to HIV DNA single-stranded with the help of the reverse transcriptase enzyme. This process by the process by which HIV RNA is converted to HIV DNA is called reverse transcriptase. So here we're saying once this HIV RNA is released into the cytoplasm, these reverse transcriptase enzymes will help convert this HIV RNA single strand into HIV DNA single strand. These HIV DNA single strands will be exactly or will be the same, because in the cell we have DNA. And this HIV, as it's coming in, it has RNA. So when it comes in, it has to convert into DNA so that they will be the same. They will have the same DNA. There will be HIV DNA and the human cell DNA. So if it was RNA and DNA, they could not combine. So the reason why the HIV RNA is converting to HIV DNA is so that they can have both DNA so that it can be easy for fusion. So this point is just saying the HIV RNA will be converted to HIV DNA by an enzyme called reverse transcriptase enzyme. And this stage is called reverse transcriptase. So the second stage is called reverse transcriptase. So the single-stranded HIV DNA then converts into double HIV DNA and enters the nucleus. So while outside there, these single-stranded DNA will now change and go into double-stranded DNA and enter the nucleus. So once that happens, it means the HIV DNA is almost the same makeup as the DNA of the cell, because it also has a double-stranded DNA. So once that happens, it means that both DNAs can now easily be compatible and mix. So after it changes to single-stranded DNA, it can now move into, after it changes to double-stranded HIV DNA, it can now move into the nucleus of the cell. So this is just talking about some of the enzymes that are released after. Just a quick recap to say, when HIV attaches to the cell membrane of the cell, it enters, and upon entry, the RNA is converted to DNA. And then there are three enzymes, which is reverse transcriptase, then there's integrase, and then there's protease. So now we are saying the last point was the HIV DNA double strand enters the nucleus. So in the nucleus, the newly formed HIV DNA is then integrated or combined with the human DNA. A long strand is formed with the help of an enzyme integrase. So once this HIV double-stranded DNA enters the nucleus, it will be integrated, it will fuse in and integrate with the human DNA and form one long strand. So the HIV DNA will now join with the human DNA, forming one DNA with the help of integrase enzyme. This allows the HIV to reprogram the human cell to make more HIV copies. So we're saying once this virus goes into the DNA, once the HIV genome, which is the HIV double-stranded DNA, enters the human nucleus, the cell nucleus, it is integrated with the human DNA and form a long strand with the help of integrase. And this is because, and this will enable the human cell to produce more HIV copies, because the human DNA is now integrated with the HIV DNA, which means the human cell's DNA have HIV DNA in them. So when they are making, when they are reproducing, or when they are reproducing other genomes or the other DNA, that DNA will contain HIV cells or HIV genome. So after that, this stage is also called the integration, or the integrase. And then we have translation. So now on translation is basically conversion from one form to another. So conversion will occur within the human nucleus. So conversion will take place in the human nucleus. The viral DNA is translated to viral RNA. So once this integration of both RNA, HIV DNA, and human DNA happens, and it reprograms the cell to make more HIV copies, there will be translation. The HIV viral DNA will now be translated to HIV viral RNA. So this messenger RNA is transported outside the host cell and then converted to the long-stranded ribosomal RNA in the ribosome in the cytoplasm. So once this RNA is now out of the cell, it will now go into the cytoplasm of the cell, and that's where it will be. So remember, we're saying once these cells integrate and form one, once this HIV DNA and human DNA integrate and form one, this will allow this human cell to make more copies of HIV. And when this happens, there will be translation. The HIV viral DNA will be translated into the HIV RNA, and this RNA will be transported outside of the nucleus, which will be converted into a long-stranded ribosomal RNA into the cytoplasm. And then from there, we'll say this long-stranded RNA DNA will need to be cut and separated into smaller copies or particles of HIV subunits. So this long strand that is produced once it comes out and go into the ribosome and be converted into the ribosomal RNA, it will need, this long strand will be needed to be cut and separated into smaller copies of HIV subunits. So the cutting and separating is done with the help of an enzyme called protease. So this protease enzyme will now cut these long-stranded RNA, HIV RNA, into smaller subunits in the cytoplasm. This will now result into thousands of these smaller particles of HIV subunits in the cytoplasm. So the process of cutting and separating into smaller particles is called budding. So budding is a process in which there is cutting of these smaller particles of this long-stranded HIV RNA into smaller particles. And these smaller particles will begin to grow and develop in an early stage. So the HIV's structural subunits, which is the cell membrane, the nuclear, the nucleocapsid, the viral RNA will combine and assemble to make up the content of a new virus or a genome. or a viron. So here we are saying these small particles that have now been broken down by the process of budding will now start assembling, making these subunits of HIV structures will now start assembling to form new viruses or new virons. So when they assemble, they will form one, not only one, but they will form plenty of viruses or virons. So maturation of the new HIV should take place before other CD4 cells are attached. So what happens here is that when the HIV cell enters in the in the in the in the cell membrane, in the cell of the human cell, and all these things that we've spoken about happen. So once this happens now, where there is budding and the new virus is produced, the CD4 cells can recognize that this cell is ours, but it is defected. So the cells of the new viruses will have to form quickly before the immune system can recognize and attack these cells. So the reason why it is not that fast, the immune cells attacking the CD4 cells, is because it is the same cell it is the same cell. It is the other CD4 cells that are infected. So once this new virus is formed in the cell, it has to mature fast before the CD4 cells are attached. Because once the CD4 cells are attached, it can cause damage to the cell and will cause the virus to be damaged and the cell to be damaged as well. So the cell that has been attacked is damaged and dies in the process. So remember, we are saying once these new viruses mature, they have to mature before the other CD4 cells attack it. So once they mature, they will cause the cell in which they have attacked, or the cell that they've attacked, to be damaged and dies. And once this cell dies, it will produce thousands of viruses into the bloodstream. So the two viral RNA strands and the replication enzymes come together and co-protein form around them, forming a capsid. So as they have now destroyed this CD4 cell, that is the body cell, it will now cause the two viral RNA and this replication enzyme and all these other things to come together and form a capsid, a protective layer outside the virus that will be able to go outside there and attack new cells. So this immature viral particle leaves the cell acquiring a new envelope of the host and viral proteins. So after the destruction of this viral particle, it will leave. After the destruction of the cell, the immature viral particle will leave the cell acquiring the envelope. So this envelope that will be composed of the cell membrane of the cell. It will also have viral proteins that are coming from the two viral RNAs, the replication enzymes, and the co-proteins that were forming the capsid. And that will form one, or even we can just say thousands of viruses that are immature. Then this virus will mature and become steady to infect other cells. So once this virus now goes out from the cell that they have destroyed, it will now become mature and ready to attack other cells. So once this happens now, you find that the virus will replicate 1 billion times per day, destroying the immune system. This suppresses the immunity, eventually causing AIDS. So now we're saying this virus or these viruses that are produced from the cells that are infected will now mature, and when they mature they will be able to infect other cells, and as they infect other cells they will replicate 1 billion times per day, destroying the immune system of the person, causing suppression of the immunity, which will result in AIDS. So this is the simple life cycle that we can talk about, and this is the diagram that we can easily summarize what we are talking about. So that is all we had on the life cycle of HIV. I hope you enjoyed our lesson. If you have any questions, you can comment down in the comment section so that we can interact. And also make sure that you subscribe to our YouTube channel and also share our videos to other people. Thank you for your time. See you next time.